Chapter 3, Inflammation and Healing. So inflammation is defined as a complex local response of living vascularized tissue to injury, such as infection or tissue damage. It brings cells such as WBCs and molecules which are necessary for host defense from the circulation to the site of injury where they are required to eliminate the offending injurious agents. So basically, whenever there is any injury or offending agent or substance in the extravascular tissue, there is recognition of that agent by the host cells, then there is emigration of WBCs and escape of plasma proteins to the offending agent where it is located. And these leukocytes and plasma proteins, they destroy and eliminate the offending agent and substance. There is control and termination of the inflammatory reaction, and there is repair of damaged tissue. So types of inflammation, you have two types, acute and chronic. So basically acute, you can remember it as DOP, duration, onset, and P for predominant cells. So acute is rapid in onset. Chronic, it follows acute inflammation or it is slow in onset. Next, duration, it is of short duration acute. Chronic is of longer duration. Predominant cells are neutrophils or PMNLs in acute inflammation, and chronic you have lymphocytes, monocytes, macrophages, and plasma cells. So characteristics in acute inflammation, basically you can see exudation of fluid and plasma proteins, so edema and leukocyte emigration. But chronic inflammation, you can see neovascularization and fibrosis. So signs, local and systemic signs are prominent in acute inflammation and less prominent in chronic inflammation. Moving on to the cardinal signs of inflammation, you have four things: rubor, calor, tumor, and dolor. So the fifth clinical sign, it was added by Rudolf Virchow. It is functional ASA or loss of function. Okay, it's a clinical sign. So four cardinal signs were given by Celsus. The mechanisms: so rubor, redness, it is caused by increased blood flow and stasis of the blood. Calor, again it is caused by increased blood flow. Tumor, it is caused by increased vascular permeability causing escape of protein-rich fluid from the blood vessels. And dolor, or pain, is caused by chemical mediators such as prostaglandins and kinins. So causes of stimuli for infection, we have causes of stimuli for inflammation, we have infections, we have tissue necrosis, so ischemia, any physical agents, trauma, thermal injury, burns, radiation, electric shock, chemical injury, foreign bodies, and immune reactions such as hypersensitivity reactions and autoimmune diseases. So first what happens is we have patterns, recognition, receptors. So recognition of microbes, necrotic cells, and foreign substances. The first step is recognition. This is done by receptors and circulating proteins. So first we have pattern recognizing receptors or PRRs. Most of them belong to the TLR family, toll-like receptor family. They're present on recognizing cells such as phagocytes and dendritic cells. And what they do is they recognize PAMs, that is pathogen associated molecular pattern. Okay, these PAMs include mannose, oligosaccharides, or peptidoglycan, or LPS, or CpG DNA. Okay. Next, toll-like receptors, they are named after the founding member toll, which is a gene that was discovered in Drosophila. So they are located in the plasma membrane and endosomes, and they are capable of detecting PAMs derived from extracellular and ingested microbes by or bacteria taken into a phagosome by phagocytosis. There are 10 expressed TLR genes in humans. So TLRs are single-pass transmembrane proteins, and the extracellular region is LLR domain, so leucine-rich domain, leucine-rich repeat, such as CD14. So what happens is they can recognize your microbes, okay, and they can send signals. So once the microbes are recognized, they stimulate the production and expression of many secreted and membrane proteins. So these proteins include cytokines that induce inflammation, antiviral cytokines, interferons, etc. So we also have sensors of cell damage. Not only for microbes, we also have cell damage sensors. So cytosolic receptors, they recognize cell damage associated with the molecules or proteins. They are called DAMPs, so damage associated molecular patterns such as ATP or uric acid or increase in intracellular potassium, etc. And this activates an inflammasome. It is a multi-protein cytosolic complex. So inflammasome can recognize products of dead cells such as uric acid and microbial products, and it stimulates the production of cytokine interleukin 1. So interleukin 1, it recruits leukocytes and there is inflammation. So gain of function mutations cause autoinflammatory syndrome. So inflammasome and associated disorders, it is also responsible for inflammatory reactions to urate crystals, so gout, cholesterol crystals, so atherosclerosis, lipids, so metabolic syndrome and obesity associated diabetes, and amyloid deposits in the brain, Alzheimer's disease. IL-1 plays a role in atherosclerosis and type 2 diabetes mellitus. Next, circulating proteins. So many plasma proteins recognize microbes and function to destroy the blood-borne microbes and stimulate inflammation. So complement, we have inactive plasma proteins in the complement system. We have mannose-binding lectin. It's a circulating protein which recognizes microbial sugars and promotes ingestion of microbes and activates the complement system by the alternate pathway. And you also have collectins. These proteins also bind to microbes and promote phagocytosis of the microbes. Next, acute inflammation. So basically we have vascular changes and cellular changes. So starting with vascular changes, the purpose is to deliver the circulating cells, fluids, and plasma proteins from the circulation to the site of infection or injury. So first we have vasodilation. It is the earliest feature of acute inflammation. You need to increase the flow of blood containing WBCs and everything to the site of injury, right? So vasodilation. So the effect is increased blood flow, local heat and redness. So the chemical mediators involved, see, remember, for increased permeability as well as vasodilation, histamine and kinins are common. Okay, but in vasodilation you have prostaglandin and endo, but in increased permeability you have leukotrienes. So prostaglandin here, leukotriene here, and then nitric oxide, obviously vasodilation. Next, increased permeability of the microvasculature. It leads to escape of protein-rich fluid from the circulation to the extravascular tissues. Again, you have these chemical mediators, and there is slowing of blood flow, which leads to rubor, and it increases the viscosity of the blood due to stasis. Okay, responsible for localized redness. Next, once you increase the flow, you need to cause the plasma proteins from the blood to leak into the extracellular tissue. So there is increased vascular permeability. So exudation is defined as the process of escape of fluid, proteins and circulating blood cells from the vessels to the interstitial tissue or body cavity. So escape of a protein-rich fluid causes edema, and it is one of the cardinal signs of inflammation. So we have differences between a transudate and an exudate. Remember pi. So protein-rich inflammation associated is exudate. Okay, non-inflammatory and non-proteinaceous, it is transudate. So mechanism is, it is an ultrafiltrate of plasma, transudate. It is caused by increased hydrostatic pressure, whereas exudate is caused by increased vascular permeability to proteins and all. So that because proteins are present, the exudate specific gravity is more than 1.018, and this is less than 1.018. And protein content, it is low, it is high here. Fibrin content again is low, and it does not coagulate in transudate, whereas fibrin content is high and it coagulates because fibrin is also a plasma protein kind of thing. And again, cell count is low, cell count is high because it is associated with inflammation. Types of cells, you have few lymphocytes, but here you have neutrophils in acute and lymphocytes in chronic inflammation. Bacteria and LDH are usually present and high in exudates but absent in transudates. So there is pitting edema in transudate and non-pitting edema in exudate because proteins are there. Next, so mechanism of increased vascular permeability. So several mechanisms are there. First is contraction of the endothelial cells. It is an immediate transient response. So it is caused by histamines and kinins commonly, and also LKT. Next, direct endothelial injury. It is caused by burns or infection, etc. It is an immediate sustained response. Then you have leukocyte mediated vascular injury. The leukocyte, mainly neutrophils, which adhere to the endothelium during inflammation, may themselves injure the endothelial cells. Next, you have increased transcytosis. So process of transport of fluids and proteins through the channels is called vesiculovascular organelle. It is increased in number during inflammation, and there is leakage from new blood vessels. These vessels are leaky till the endothelial cells mature. So you have damage to the cells, you have contraction of the endothelial cells, you have leukocyte mediated vascular injury and leakage from the blood vessels, and increased transcytosis by vesiculovascular organelle. Next, the cellular event. See, we need to remember that increased vascular permeability especially occurs in the post-capillary venules. Okay, in lungs it occurs in the capillaries. Next, cellular events. This delivers leukocytes to the site of injury. These neutrophils are produced in the bone marrow. They are rapidly recruited to the sites of inflammation and are short-lived. So macrophages are slow to respond but long-lived. Leukocyte events can be divided into leukocyte recruitment and leukocyte activation. Okay, so if you were to divide the phases of inflammation, first you have the fluid phase, then you have the acute inflammatory neutrophil phase, and then you have the macrophages. Okay, chronic. Next, steps in leukocyte recruitment. So basically it is defined as recruitment to sites of inflammation is a process of migration of leukocytes from the lumen of the vessel to the site of injury in extravascular tissues. So journey is a multi-step process. So in the vascular lumen, normally leukocytes move rapidly in the flowing blood, which prevents them from attaching to the vascular endothelium. In inflammation, they should be stopped and then brought to the offending agent or the site of tissue damage outside the vessels. So first you have margination. So normally they are flowing in the blood, but they need to come to the peripheral wall of the blood vessel, so that is margination. So when the blood flow slows down or there is stasis, leukocytes move towards the peripheral column. They accumulate along the endothelial surface. This is called margination. Then there is rolling. So margination, once the leukocytes come to the endothelium, they're able to detect and react to changes in the endothelium. They attach weakly and then dissociate again, and then there is a jumping movement which appears as rolling. So the molecules involved in rolling are selectins, whereas molecules involved in adhesion, once the rolling stops and the leukocytes adhere, they are integrins. Selectins here, integrins there. So selectin family of adhesive molecules are involved. So selectins mediate the initial weak interactions between leukocytes and endothelium. So IL-1 and TNF are the chemical mediators involved. Okay. So E-selectins, endothelial selectins, are activated after stimulation by cytokines and other mediators. So you have LEP. Okay, L is present on leukocytes. So L-selectin is present on neutrophils, monocytes, etc. And then E-selectin and P-selectin, they are present on endothelium. They are activated by cytokines such as TNF and IL-1. The ligand on everything is silyl Lewis X. But only thing here, because the L-selectin is present on leukocytes, the ligand is present on glycam-1, whereas the silyl Lewis X in both of the other things, E and P-selectin, is present on leukocytes because this is present on the endothelium. Next, addition of leukocyte to the endothelium. The endothelium finally gets activated and leukocytes bind more firmly and finally come to rest at some point. They assemble as pebbles over the endothelium. This is called pavementing. Next, molecules involved are integrins and corresponding ligands. So the chemical mediators involved are again TNF and IL-1. So drugs that block TNF leukocyte recruitment are used for the treatment of chronic inflammatory diseases. So chemokines, they cause conversion of low integrity integrins, low affinity integrins on the leukocyte to high affinity state, resulting in firm adhesion of the leukocytes to the endothelium. So first there is margination, then there is rolling, and then there is adhesion. So types of integrins, you have beta 1 and beta 2 integrins. So beta 1 integrins, they are located on the monocytes and T cells. See, all of them are on monocytes and T cells. Okay. Whereas the complementary ligands, basically for beta 1 it is VCAM, and for this one it is ICAM. So vascular cell and cell adhesion molecule and intercellular adhesion molecule, VI, beta 1, beta 2. Next, once it adheres, then it can transmigrate, right? So there is transmigration or diapedesis. So leukocytes migrate through the vessel wall by squeezing through the intercellular junctions between the endothelial cells. The molecules involved are PCAM1. So it includes a member of the immunoglobulin superfamily called CD31 or PCAM1, platelet endothelial cell adhesion molecule. So leukocytes penetrate the basement membrane by secreting collagenases. Outside the vessel wall, there is chemotaxis. So it is defined as a process of migration of leukocytes towards the inflammatory stimulus in the direction of the gradient of locally produced chemoattractants. So what are these chemoattractants? You need to remember 4 things. So non-bacterial products, you have leukotriene B4, C5a, IL-8, and then bacterial products, you have exogenous ones, endogenous, exogenous, leukotriene B4, C5a, IL-8, and bacterial products. Next, there is accumulation of leukocytes at the site of infection. So once the chemokines attract the leukocytes after they went out of the vessel, then leukocytes accumulate at the site of infection. Okay, then they bind to the extracellular matrix proteins through integrins and CD44. Okay. Next, types of leukocytic infiltrates, neutrophils predominantly during the first 6 to 24 hours, then monocytes. Then clinical importance of leukocyte adhesion molecules, you have 3 types, so LAD1, LAD2 and LAD3 deficiencies. They are transmitted as autosomal recessive diseases. So it is characterized by the inability of neutrophils to exit the circulation to the sites of infection, leading to leukocytosis and increased susceptibility to infection. Okay, so basically Pseudomonas infection, neutrophils predominate over 2 to 4 days also. And genetic deficiencies of leukocyte adhesion molecules cause recurrent bacterial infections and also leukocytosis, because WBCs, the body thinks that they are not enough to kill the organism. Next, so there are integrin defects in LAD1, and LAD2 you have selectin defects, and LAD3 you have firmed 3 mutations. Okay. Next, LAD2 is associated with Bombay blood group and has mental retardation, and LAD1 it is associated with delayed separation of the umbilical stump. That is very, very important. Next, leukocyte activation. So once the leukocytes accumulate at the site of injury, then there is recognition of microbes of the dead cells by the leukocyte receptors, which initiate several responses in leukocytes together known as activation. The most important ones are phagocytosis and intracellular killing. So first what happens is, phagocytosis consists of 3 steps: recognition and attachment, next engulfment, and intracellular killing. So first, recognition and attachment, it is done by various receptors. You have toll-like receptors, G-protein coupled receptors, cytokine receptors such as interferon gamma, mannose receptors, scavenger LDR receptors, and opsonin receptors. So receptors for opsonins, the phagocytosis is enhanced when leukocyte receptors recognize microbes coated by specific host proteins known as opsonins. These proteins could be IgG antibodies or the C3b breakdown product of complement, and certain plasma lectins, especially mannose binding lectins called collectins. So the clinical significance of opsonins is that after exposure to the antigens, B cells get activated and mature into plasma cells, which produces IgGs, which acts as a opsonin. So Bruton's disease, there is X-linked hypogammaglobulinemia. So there is a defect in maturation of the B cells. So pre-B cells are there, but B cells are not there. It leads to absence of immunoglobulin production. There is defective opsonization in Bruton disease. Next, engulfment. Once it recognizes, it needs to engulf. So the phagosome, it's the extension of the cytoplasm of the leukocyte forms pseudopods surrounding the particle to be ingested, and it forms a vesicular vacuole called a phagosome. Then it fuses with the lysosome to form a phagolysosome, and lysosomal granules are discharged into this phagolysosome. Next, during engulfment, the phagocyte may also release some of its contents of its granule into the extracellular space, and this may damage the innocent normal cells. So, clinical significance in phagolysosomal function, we have Chidiac-Higashi syndrome, which is due to defective phagolysosomal fusion. So, there is increased susceptibility to infections, and there is neutropenia, there is decreased numbers of neutrophils, and defective degranulation. So, that is why in peripheral smear you can see leukocytes with giant granules due to aberrant phagolysosomal fusion, and you have mutations in the LIST gene here, the lysosomal trafficking regulator, which regulates lysosomal trafficking. And also you can see albinism due to abnormalities in melanocytes, nerve defects, and bleeding disorders. Next, there is killing and degradation of ingested microbial agents or particles which occurs within neutrophils and macrophages. The most important ones are reactive oxygen species, reactive nitrogen species, lysosomal enzymes, and granule enzymes. So first, what happens is within the phagolysosome, what happens is NADPH is converted to NADP by NADP oxidase, and in this process, oxygen is converted to superoxide anions. The superoxide anions, they undergo spontaneous dismutation to form hydrogen peroxide. Hydrogen peroxide is insufficient in that amount it is produced to kill most of the microbes by itself. But the enzyme myeloperoxidase converts the hydrogen peroxide into a powerful ROS. In the presence of halides such as Cl, it converts hydrogen peroxide to HOCl, which is hypochlorite, and that is the active component of bleach. So it destroys the microbes by halogenation and lipid peroxidation. So oxygen derived radicals may be released extracellularly from leukocytes and may produce tissue damage during inflammation, and serum tissue fluids and host cells, they have antioxidant mechanisms. Reactive nitrogen species, so nitric oxide is a soluble gas. It is produced by from L-arginine by the action of nitric oxide synthase, so NOS. It can kill microbes similar to ROS. So inducible NOS type is involved in microbial killing. INOS is expressed when macrophages are activated by cytokines or microbial products. The INOS produces nitric oxide within the macrophages, and this nitric oxide reacts with superoxide and forms ONO, which is a free radical peroxynitride. It also causes peroxidation and attacks the microbes. Next, lysosomal enzymes. Within the lysosomes, you have acid hydrolysis, which degrade the dead microorganisms. Elastase can kill bacteria. And also granule enzymes. So neutrophils and monocytes have granules, primary and secondary granules containing enzymes and antimicrobial proteins. The primary granules are large, and they have non-specific azurophil granules, whereas the secondary granules are small and they are specific. Monocytes, again, they also have granules. So basically, phagocytic vesicles containing engulfed material may fuse with the granules of leukocytes and also with lysosomes. They'll destroy the ingested material. If the initial leukocytic infiltration goes unchecked, the enzymes can damage the normal tissues also. This is normally controlled by antiproteases such as alpha-1 antitrypsin, but deficiency of this may lead to sustained action of these proteases and damage the tissue. Moving on to neutrophil extracellular traps. So during the formation of net, there are extracellular fibrillar networks produced by neutrophils in response to infectious pathogens. So during the formation of that, the nuclei of the neutrophils is lost. It leads to netosis. It is a distinctive form of death of the neutrophils. Nuclei is lost. So composition net consists of a viscous meshwork of nuclear chromatin that binds and concentrates the granule proteins such as antimicrobial products and enzymes. So they concentrate the antimicrobial substances at the site of infection, and they prevent the spread of the microbes by trapping them in the fibrils. It is a non-phagocytic mechanism of killing the microbes. So nuclear chromatin in the nets may be a source of nuclear antigens in systemic autoimmune diseases, so that is a significance, such as in lupus. Then you can have inherited defects in microbicidal activity. So you have chronic granulomatous disease, so CGD. It is a group of congenital disorders characterized by defects in bacterial killing. So there is decreased oxidative burst. It is due to defects in the genes encoding and NADPH oxidase, phagocyte oxidase, which generates superoxide anions. So X-linked defect could be there, and autosomal defect could also be there. Next, the people are susceptible to recurrent bacterial infection. It is named granulomatous because the initial neutrophil defense is inadequate, and there is chronic inflammatory reaction rich macrophage that tries to control the infection. Next, diagnosis is done by nitropruritazolium reduction test, so NBT test. This depends on direct reduction of NBT by superoxide anion to form an insoluble formazan. It is positive in normal individuals with normal NADPH oxidase, but negative in CGD. You also have dihydrorhodamine test. So whole blood is stained with DHR, incubated and stimulated to produce superoxide anion, This free radical reduces DHR to rhodamine in cells with normal NADPH oxidase. And you also have cytochrome C reductase assay and myeloperoxidase deficiency. There is decreased microbicidal killing because of defective MPO-H2O2 system. Then you have acquired defects of leukocyte functions, so decreased production of leukocytes, defect in leukocyte adhesion and chemotaxis, and also defects in phagocytosis and microbicidal activity. Finally, there is termination of the acute inflammatory response. So it is terminated after the elimination of the offending agent. It is brought about by the following. So acute inflammatory process is reduced after the causative agents are removed. This is because the chemical mediators of inflammation, they are produced only as long as the stimulus persists. They have a short lifespan and are degraded after their release. Neutrophils also have a short lifespan and die by apoptosis within hours to day or two after leaving the bloodstream. The cells themselves stay, the chemical mediators are short-lived, and also there is variety of termination mechanisms. So there is a switch in the type of metabolite produced from arachidonic acid, namely pro-inflammatory leukotrienes, to anti-inflammatory lipoxins, and the liberation of anti-inflammatory cytokines such as TGF-beta and IL-10 from macrophages and other cells terminates the response.